Okay, so hi everyone. Thank you very much for joining the SCSE Club today. So I'm Aaron from Bioturing team, and today I will share with you about the new Talk2Data version that we recently we have released. And and with the new the talk to data, we can analyze we can perform the analysis across the database with the significant increase in the speed of analysis, and we also have the expanded in database and also in species as well. And in this version of the talk to data, we have the AI function integrated, so it can help with discovery of the research question using AI function. And this is the quick agenda for today. So I will go through some what is the new in terms of data structure that behind the new talk to data, and then I will show you what is the databases that we will have in the new talk to data, and also the species, which is we have more species compared to previous version, and option to use ASI agent, and we also have the BioFlex SDK, which is the convenient way to integrate the talk to data analysis and also the data from talk to data to your pipeline. And I will go through some study exploration using either in individual study or we can do like cross database analysis. And also in this also demonstration how to use the AI function to search and also to analyzing data using AI Sonya, which is built by Turing. And also I will introduce a little bit about the Disease Explorer program. Okay, so let's get to that. So first about the database. So as we can see, so the single cell and spatial transcriptomics and also bulk RNA is expanding day by day. So so we can see that the traditional database is no longer be um, can adapt with that. So the expansion of the single cell data is causing so the limitation of a traditional database. So the database gonna get like bigger and bigger, but with the traditional one, the database the analysis become like getting slow, and with the traditional database we cannot improve that. So for the new talk to data, we develop a new data structure and combined with the massive parallel computing to able to analyze massive scale of data in real time. So it is the very different with the traditional database. For example, here we can query a gene across the whole database, and we can able to show the expression of this gene in with in individual study at in real time. When you query for a gene, it will show immediately in real time. So that is the very significant difference compared to the traditional database, which is here is done in real time. Okay. And with the expanding of the database bases and species, so with the new talk to data, so the previous version that we have is species human and database is single cell. And with the new version, we will have single cell, bulk RNA, and also spatial transcriptomic gonna become very soon integrated into the database. And in terms of species, we will have human, mouse, and non-human primate, and we're planning to expand more in terms of species to the database. And you can find the information in the interface here, which I will show you very soon. Okay, and the one important feature that we have in the new version of the Talk2Data is the AI integration into the Talk2Data. So you can see here we have Sonya, which is the AI model that we train on for biomedical research, and it's also trained on the Talk2Data database, so they can able to um, answer the question for biomedical researcher, and also able to search for the study that you interest, and also able to analyze the data from the database as well. Okay. And the second model that we have here is we call Hera. So Hera is the model that we based on Sonya, but with that model, Turing scientists can able to build that and custom that for researcher based on their in-house data and also for the custom data set. So if you have your in-house data and you would like to build a model that AI model that's able to analyze or understand your in-house data, we also able can do that using Hera model here. And beside that, we also have the option to integrate your own model into the Talk2Data. So if you have the researcher already have their own AI model and they want to integrate in Talk2Data, for some here we can have some cloud model, and you can able to integrate your model into the Talk2Data platform as well. Okay. And also another feature that also new in this version is the BioFlex SDK, which is instead of using UI user interface to do analysis, so now user can able to run command line to do the same analysis that you can do with the user interface. So one of the benefits of the BioFlex SDK is that the output from the analysis can be entered directly into the user pipeline. So it can be integrated into the pipeline, and then we can have the seamless workflow from that. Okay. And in the future, the BioFlex SDK will be used to integrate different modality from the database to kind of unify for the analysis. Okay. So now let I go to the user, the Talk2Data, and then we can do some analysis using the new version. Okay. So this is the user interface for the new Talk2Data, and you can see here the list of study, and we have database here, and we can also able to go back to the previous version as well. So if you would like to go back to the version 5, version 4, we also have the option to do that. Okay, so first with the user interface, the first one is the study tab here. In the filter panel here, we have like database. So now we have single cell and bulk RNA, and in this month we also planning to add the spatial transcriptomic data into this database so we can expand the data. And in terms of species, we have human, mouse, and non-human primate for your reference. And we have also different data set for some human cancer, say human cell atlas, which is currently we make it published for everyone to able to access the data, or the whole 100 and so publicly available for everyone to try. And we also have the Sonya version here. Okay. So I think there are four ways for you to for people to able to explore the data on the new Talk2Data version. So the first one is we can analyze individual data. For example, here we can go study, and then we have the list of study here. And for example, if I interested in breast cancer, so I can filter um, for the study, which is similar at the previous version. And for some condition here, I can do the breast cancer. Okay. And the treatment, for some, I interested in anti-PD-1 treatment. And we can see we filter out for 6 study. But in this version, we have the option to easier to look into the study of your interest. For example, we go here on the setting, and then we can select different parameter to show here. For example, I interested in the treatment and whether they have any response on treatment, for example. Okay. So from that, we can look into have the overview of what is the criteria that I want to have, and what is the whether or not this study have that criteria. For example, if I interested in the anti-PD-1 treatment and I would like to have like non-response and response condition, so I probably go to this study because other study they don't have that information, for example. And then I can go here, and then I can click explore. So the study will be open using the P-Browse Explore, which is the platform from Battery that specialize for single cell data analysis. So you can able to continue more do more analysis on using B Process Pro. And I believe in this series, the SCSE Club, we have many video on how to using B Process Pro to analyze single cell data. So please feel free to um, go to this video for the analysis. And so I can focus more on the new Talk2Data. So that is the first way that we can analyze Talk2Data version data on Talk2Data. So the second way that we can do here is we can do the cross database analysis, which is for example I interested in analysis. For example, I interested in the brain right now. So filter brain. And then I filter condition. For example, I'm interested in normal brain and also in Parkinson's disease. Okay, and in terms of cell type, I'm interested in dopaminergic neuron. Okay. So from now, with the new interface of the Talk2Data, we can do the analysis seamlessly on one page. We don't have to switch between different tab or different panel for that. So for example, here I have brain, I have dopaminergic neuron, and then we can go to the tree, custom tree view here to able to set up how we can view the data. For example, the first one is anatomy, which is brain. Second one is cell type, which is dopaminergic neuron. And I want to have another level, which is condition. So we have like normal and Parkinson's disease. So now we can have all the information on one panel. And one thing, for example, I want to know what is the marker gene for the dopaminergic neuron in the brain. So for new data, it's very simple. We just click into that neuron, and the platform will calculate in real time the top 100 marker gene for you. And you can see the list of marker gene here for some like tyrosine hydroxylase here. And from that, you can able to see the expression of the gene by just simply click into the gene, and you can see what is the expression of gene in this condition. Okay. And another thing that you can do on the same user interface here is, for someone I interested in knowing what is the differential gene expression between the Parkinson's disease and normal in terms of dopaminergic neuron. So what I can do, so I can go here in differential expression, I select that. The group one, I select like Parkinson's disease, put to group one, normal group two. And then from that, I just run the analysis. And now the platform run the real-time analysis, and you can see they immediately show you the list of differential gene expression here, and you can select some gene to see what is the expression level between these genes. Compare between the Parkinson's disease and the normal condition. And for someone that would like to understand more about this gene, for example, I run X1, and I want to know what is the gene that causes with run X1. So what I can do is I can simply click into the run X1 and in the Parkinson's disease condition, and then the platform will calculate the gene that show you they will show the top 10 gene that correspond with run X1. So you can see within one single user interface, we can narrow down for the cell and the condition and the tissue, and then we can do differential gene expression, and then we can do the co-expression at the same time. So it's a very convenient way. So it will help with the analysis, quickly read the result. Okay, so that is the second thing that we can able to do with the new platform. And with this platform, we have different option here. So let I clean this. So we can view the expression using different methods. For example, is here. I can go to run X1. And then you can see the expression level of run S1, and we can able to see the expression of run S1 at different condition. That I remove this. computer. So it will show the redesigned expression between different conditions shown here, different tissue. Go to analysis here. And then you can able to sort the expression of the next one. You can see which tissue is highly expressed for the next one. And you can do the same thing with the cell type. You can have the list of all cell type, and you can identify which cell type that highly expressed for the next one, for example. Okay. So for that is the second way that we can explore the database, which is we can do the analysis across the database. And the third option here that we can do is we can use AI to help us to do the analysis. One of the, I think it's very interesting and nice ways to do. So for example, I asked a question. So for AI, we have different model, but I will use the Sonya, which is developed by Baturin here to demonstrate that. So we have different options. So for example, here is ask, which is using the LLM to answer your biological question. For example, if I would like to ask, which gene are commonly used as a drug target for treating colorectal cancer, for example. So I ask that question. So the AI Sonya will use the LM to answer to give you some answer for what is the most commonly used as a target for drug development of colorectal cancer. And then for example, you can click into the gene and then you can see which data that we have in that in the talk2data database. Okay. And the next question, and the second option here is agent, which is enable to help you to do the analysis, not just answer some question based on the LM, but you can able to do analysis as well. So for example, from here, I'm interested in BRAF. And the next second question that I would like to ask is, which gene that co-express with BRAF in the colorectal cancer condition? So first, the Sonya will give you kind of little bit of overview of the BRAF and also potential gene that co-express. But then the Sonya AI will thinking on the database, which is they select the tool and perform the analysis, actually based on the data on talk2data. So that is the very different compared with the other AI that we have. So they first showing you the step that they kind of reasoning and what is the step that they're gonna do. And they will show you the step they do and what is the tool that they do. So kind of like put the clear context of what the analysis that they do. It's not in a black box, but they will show you in the real context here. Okay. And then they will summarize the key finding of subgene that co-express with BRAF. And then you can go to further analysis with these gene. And in the future, what we can do using this agent mod here is we can able to see the draft that actually draw from this agent. So for example here, I can the researcher ask the AI Sonya research question, and then research AI Sonya do reasoning and select method and tool to do analysis, which is I just as the one I just showed you, and then they actually able to display the figure for analysis, and then finally they can able to summarize the key finding for the researcher. So instead of using the UI to do click and drag different conditions, so if you can able to use your natural language to able to analyze data on the talk2data database. Okay. And the last option here is you can search for the study. For example, if you for example or you give you some gene that co-express with BRAF in colorectal cancer, and then I would like to search for the study that have that gene, for example. So I will use the option to search for study on Bacterial database. So list on of study with breast cancer, and for example is EGFR. And first, then, so give you the overview of EGFR in breast cancer, and then they will show you the list of study that actually be stored in the Talk2Data database. So from that, you can further able to explore the data. Yeah, so they list the on of a study that you can see on the Talk2Data. And then so in the next release, you can able to see the similar list of study with the Talk2Data that we see on the study tab. And then you can able to click into this study and go to further explore using VFR sites, for example. So from that, it's a seamless workflow from the question, the research question that you can ask. So to help with to raise any idea about how to find a target for drug development, and then we can identify there are already some target for drug development, but we would like to find for some new target for drug development, and then we can using the code expression to identify any potential new drug target, and then you can able to search for any publication related into that new drug target, for example. So that is the seamless workflow using the AI. And one last thing that I would like to mention here is a fourth thing that you can analyze the data on the Talk2Data that I just mentioned in the beginning, which is the BioFlag SDK. So with BioFlag SDK, you can install this for free. It's come with the Talk2Data. And then you're able to do analysis using your command line. And as I said, for the output from this analysis, you can integrate that into your pipeline. So instead of going to the user interface, you can tie using the question using Sonya AI. We also have another option for you to able to directly integrate the Talk2Data and the Talk2Data database to your pipeline in the analysis as well. So I think for that, I cover most of the new function for the Talk2Data. And if you have any more question, so so please feel free to reach out to us. We have the support email, so you can reach out to that for more information or more in-depth training on that. And one last thing that I would like to mention is about the ADC Explorer program. So with the new Talk2Data, we are expanding the data set for the whole database. And from that, we also, because with the advance of the new a new database structure and the massive parallel computing, we now able to do some custom DC Explorer or custom atlas for the Talk2Data. For example, if a researcher interested in particular data set or particular disease, we're able to customize that and to put into the Talk2Data platform. And from that, we can do the file curation on that. So it means we're able to select the particular study that fit with the result of interest, that think that is the study that the researcher think that is the high quality, and integrate that into the atlas. And from that, we can able to the researcher can able to analyze the data using that atlas only. They can do the cross study analysis using that atlas only, but not the whole database at the new Talk2Data. Okay. So and with the new Talk2Data, we very exciting, and we also put some public data set, so everyone can able to explore that. For example, you can explore the public data set from human cell atlas and the Tahoe 100 million. Using this link, you can go to talkdata at bioengineering.com, and you're able to freely access into the two datasets, and feel free to explore with the new function of the Talk2Data. And I think from that, um, we can there are some resource shown here. So you can able to go to new Talk2Data documentation, which is actually in the Talk2Data platform, and then you can watch tutorial video about Talk2Data as well. So I think so now we have some question from the poll. So you can able to uh, vote for that. And if you have any questions, so please feel free to put into the Q&A or in the chat box. Okay, so let us look into chat box here. Okay, so I put in the chat box uh, the support email if anyone interested in using the new Talk2Data or would like to explore any further about Talk2Data. So please feel free to reach out uh, to us. And if you have any question that's related into the new version of Talk2Data, so please feel free to drop the email as well. Okay, I think yeah, I think our college just drop up the email to the chat box, so please feel free to submit any request for that. Okay. Okay, so maybe I just give a few minutes for the poll to finish. And thank you very much, everyone, for joining the section today. And I hope it's a useful section for your research. And hopefully, you can explore all of these new feature of Talk2Data and benefit for your research. Thank you very much for joining the SCSE Club today.